Okay, thanks everybody for joining us this afternoon. It is indeed a virtual seminar, but I'm coming to you from an auditorium because we have a few students joining us and viewing it from here. This is our second seminar of our Timothy A. Johnson Medical Scholar Seminar Series. This is a series, as many of you know, that allows both the VTC School of Medicine and the Fralin Biomedical Research Institute to bring in speakers like Dr. Neil Hanchard, who we're going to hear from in a minute, who are practicing physicians, but also building their research careers based on their interaction with medicine. And so it's a powerful series that is named after the first Dean of Research here at VTC-SOM, and he helped us envision this series, and we are grateful to his inspiration for this particular seminar series. Today, we're going to hear from Dr. Neil Hanchard, who earned his MBBS from the University of West Indies in Kingston, Jamaica. And then he received a pretty pivotal Rhodes Scholarship, as I'm sure many of them frequently are. But in Dr. Hanchard's case, this led him to Oxford, where he ended up earning a PhD in clinical medicine, specifically human genetics. And while there, when he was doing his PhD, he looked at genetic influences on susceptibility to infection, particularly in children. And he did this under the supervision of Dominic Kwiatkowski in the Institute of Molecular Medicine. And this was a bit of a departure from his original intention, which was to go into neonatology. So after this work in his PhD, he went back home to Jamaica to do a postdoc at the Tropical Medicine Research Institute. And this time he focused on population genetics of the sickle allele in the Jamaican population. He then went on, he left the warm environment of Jamaica and spent a few cold years at the Mayo Clinic, where he completed a residency in pediatrics. And then he moved on back to the warmth of Baylor College of Medicine in Houston to complete his residency in medical genetics. And then he stayed on there as an assistant and associate professor at BCM's world-renowned Department of Molecular and Human Genetics, where in addition to contributing to the very active clinical service at that department, he continued to grow his research portfolio and was also directing the very busy core laboratory for translational genomics that BCM is widely known for. Just recently in January, Dr. Hanchard actually moved a lot closer to us here at Virginia Tech. He moved to NIH, where he currently serves as head of the Childhood Complex Disease Genomics section in the National Human Genome Research Institute. And so for those of us who are particularly interested in this type of research, it's exciting to see Dr. Hanchard in a position like this, because it's going to allow him to pull his relatively diverse research portfolio together under this umbrella of childhood complex diseases. Dr. Hanchard has authored or co-authored over 100 peer-reviewed articles that explore everything from contributions to the Undiagnosed Diseases Network to the art and practice of patient communication with patients who have complex genetic disorders, as well as detailed descriptions of novel variants that contribute to human disease. Today, however, he's going to share with us his work to fill the gap that exists in our full understanding of African genomics. So I'm very excited today to welcome Dr. Neil Hanchard. Thank you for joining us.
Thank you. It's wonderful to be here. I'm sorry I couldn't be there in person, but I've had a fantastic visit to this point, and look forward to continued interactions, especially now that we are so much closer. So with that in mind, I thought today we'd sort of walk through some of the work we've been doing in looking at genomics on the African continent. And so I'm going to try and walk through that, sort of make the argument as to why I feel this is important, and then use two examples to try and back that up. But I actually wanted to start by talking about our general approach to disease, which as a physician, I often start with the patient. So we start with the disease or the clinical phenotype. And from there, we think about what the epidemiology and demography of that disease is. And so from a genetic standpoint, we can have diseases that are relatively uncommon, but have a very large genetic effect. And that goes all the way to the other end of the spectrum, which is the opposite. But understanding that epidemiology and demography allows us to decide what genetic models are the right ones to employ. And so once we have a model in mind, then we can recruit these large cohorts. We can use some of the modern day technologies that we have been using to approach these disorders recently. And then that starts the nidus of discovery. And that's mostly what I'm going to be talking about. That's mostly what we've been doing. But the discovery is but a funnel, is the top of the funnel for us to do subsequent functional validation, for us to understand more about the mechanism, and then ultimately, hopefully, to have a targeted intervention, which would then close this circle of going from bedside to bench and then back. So we are doing this using all these sort of multi-omic modalities across a range of diseases in the childhood space. And what you'll notice is that they're all much more common and more prevalent in populations of African ancestry. And whilst this was not by choice, this is sort of serendipitous, it has sort of guided some of my future studies. And it's a problem because there is a diversity problem in human genetics in that the large proportion of studies of genetics, traits, and diseases have really focused on populations of European ancestry, which you see here in this Nature Review, where the vast majority of genome-wide association studies, which have been the primary drivers for sort of understanding for this discovery phase of genetic susceptibility and risk. And you can see that the vast majority have been in European ancestry individuals. And then if you break it down by individuals, you can see that African populations are only 2%. This is also true, and you look at very large databases that include sort of hundreds of thousands to 500,000 individuals who have been sequenced, and we have a feeling of their genetic variation that, again, African and African American individuals are somewhat underrepresented. And this is unfortunate because the majority of new variation in the genome is found in African populations. So this is from the 1,000 Genomes Project. And you can see here just sort of the number of variant sites per genome. African populations have far more than most of the other populations. And this is because modern humans are thought to have their origins in Africa, migrating out many years ago to populate sort of the rest of the world. And the important part about this, however, is that not everybody migrated. So there's still this large pool of genetic variation that exists on the continent. And so this is reflected in the sort of genetic variation that Africans have this very deep ancestral tree. So if you sort of go back to before the migration, these African populations have more diversity, which we've seen. And so that was very much the nidus for the development of the Human Hereditary and Health in Africa, or H3 Africa Consortium. And this is an NIH and Wellcome Trust, and more recently, the African Academy of Sciences funded research consortium, really designed to empower research and researchers on the African continent by providing infrastructure, both human infrastructure, but also sort of practical research infrastructure, by filling links between researchers and encouraging collaboration. And this sort of developed in this kind of hub and spoke way, where the consortium, there are a number of projects that are collaborative, where each one of these little collaborative spokes will be comprised of one project or one disease, but multiple projects. There are individual research projects. There are ethical, legal, and social issue projects that are part of this. And then there's a very robust bioinformatics network that's all across the continent. And they also include sort of bioinformatics training. And then there are 3 biorepositories across the continent, which are designed to take any and all samples, or at least aliquots of those that are taken from any of these research projects. And the key part about this consortium is that the principal investigators had to be from the African country where the grant originated. So this very much rooted the science in the projects of the consortium. And so now, towards the end of the first 10 years of this project, you can see that There are a number of sites across the continent that have been involved in this. There's some 34 different countries, 51 projects. There are a number of policies and guidelines that have been developed as a result of this. And many of these are African specific, but a large number are sort of relevant in the global context. And the number of consortium members has grown exponentially. And this is sort of the last time we actually had the opportunity to meet together in Tunisia. And I promise you, it's a little bit of Where's Waldo, but I am somewhere in the back here as part of this. And so currently there are well more than 70,000 research participants who've been recruited to this. Often this recruitment occurs in somewhat inhospitable environments. Sometimes that's from natural things like floods, but sometimes it's from man-made things like traffic in Kampala. And they span the full spectrum of disease. So there's everything from cardiovascular, non-infectious diseases to, of course, infectious diseases, and then everything in between. And so when the consortium started, this was the landscape of sort of whole genome sequencing across the continent. And most of this was sort of centered upon the groups in Nigeria, the Yoruba and the Igbo, who for a long time were sort of proxies for Africa, or at least West Africa. And so it was felt that in order to support the many projects that were occurring, there was a need to sort of broaden our understanding and reference for what the genomes across Africa were. And so each of the principal investigators was asked whether they were able to contribute samples to a larger whole genome sequencing effort. And then the result of that is a larger expansion where there was sort of very deep sequencing in these 7 countries in red, and then sort of lower level sequencing done in the countries in green. But overall, it's now expanded. So now we're talking about 400 individuals in total. There are some 50 ethno-linguistic groups, and then 13 different countries covered. And so within this context, one of the first things that people were interested to know is like, how does the genetic variation sort of sift itself out? And to do that, we looked at principal components analysis. And it's sort of helpful to have the mindset that in a broad sense, people often think about ethno-linguistic groups in Africa as being Afro-Asiatic or Nilo-Saharan or Niger-Congo. Niger-Congo is the largest speaking group. And then there are the Khoi and San hunter-gatherer groups. And actually, when we looked at the first 2 principal components here, we could basically separate out these Nilo-Saharan groups here in the east from the Niger-Congo groups here on this side. And then the second principal component basically described this cline that went from sort of northwest Africa through Central Africa to South Africa. And in fact, there's a very strong correlation here with geography, which we were able to see. But it's also important to note that, in a lot of these cases, these aren't discrete clusters, right? To sort of like bleed into each other as you go from north to south. But there were these outliers, one of which is this group from Nigeria. They're called the Birom. And you see that they're separate from the other West African groups. And when we look here at their admixture, so here, each line here is a different individual, and the color represents their sort of ancestral contribution. And so you see the Yoruba and the Igbo from Nigeria. You can see that the Birom, relative to these groups, have this sort of dark blue ancestry. And as we looked around the continent, the other groups that had this sort of very dark blue ancestry were in the eastern part of the continent. And through a lot of different sort of genetic manipulations, we're able to show that this dark blue is indeed the introgression of East African ancestry into the Birom. And so the Birom are a very large group. Some 2 million individuals constitute the sort of indigenous Birom. And so if you think about it, we often use these groups up here, the Yoruba, as our sort of proxies, but they may not even be good proxies for Nigeria, much less for all of West Africa and all of Africa, because of this kind of complex ancestry. So we can look at that ancestry in the same way, that admixture all across the continent. And you see this going from west through central and then down into southern Africa. You can see the variation and the clines within those variations. And this actually had a bearing upon novel variation that we were able to observe. And you see that here we discovered some 3 million novel single nucleotide variants. And these are ones that are not in any of the public databases. They've never been reported previously. And this correlated well with groups that just haven't been very well studied. So you can see here the BROM from Nigeria, again, having a fairly high number. But you can see variation within these ancestry groups. So in Cameroon, this sort of gold group here have a very similar background to the Benin and the Yoruba. But once you go outside of those groups, now you start to get much more novel variation. So each shade here is a different ethnic group within a country. Well, of course, what was most marked here is the Botswana, where there are some, like on average, about 12,000 novel variants are being discovered. And the Botswana are in southern part of Africa, and they have a large swath of this kind of like pink ancestry here. And that pink ancestry correlates with the Khoi and San hunter-gatherer groups from the southern part of Africa, and they are thought to be one of the oldest indigenous groups on the continent. And relative to these other groups, the Khoi-San group has not been well sort of sampled at the genetic level. And that shows where you see all of this novel variation. And in fact, we could quantify that in a bit more, where we could look at novel variants and we just look at the proportion of Khoisan ancestry that you have, and you see this very strong correlation. The more Khoisan ancestry you have, the more novel variants that we observe. This is also true when we looked for the sort of rainforest groups, which is formerly known as pygmies. In Cameroon, that's the dark gray. And again, if you just plot the proportion of their rainforest ancestry against novel variation, again, you see this very strong correlation. And in fact, if we just said, what's life like if we take any group that's not in that Niger-Congo group, so that's the largest group that we have, and then plot that against the number of novel variants, that once we get outside of the group that's been sampled a great deal, we start to see this very strong correlation with novel variation. Great. So we wanted then to ask, well, how much more is there really to discover? And so one way of looking at that was to start from the Botswana group and just ask for each group that we add, how many novel single nucleotide variants do we observe? And you can see that as we go up here, this is, doesn't really have an approaching plateau. But you could say, well, in each of these populations, there's going to be somebody who just randomly has a variant, and so that might be driving it. So we took out those individuals, and so we're now talking about variants that are going to be pretty common within these groups. And even though this looks like you're sort of getting to a kind of plateau here, there's still 6,000 variants that are being observed as you go from the group in Benin to the group in Mali. So overall, still a lot more variation to be found within these groups. So then we wanted to give some context to this. And one thing we thought about doing was to bring it in the context of those variants that are expected to have a damaging effect upon the protein. So these are so-called loss of function variants. And the idea is that because they cause loss of function, they're expected to be quite rare in the population. And so this is described as being subject to purifying selection. And sure enough, if we looked in all the populations and we just plotted the number of loss of function variants versus the number of variants that are expected to be benign to the resulting protein, you can see that there are far more in this group of just one. But also, we could see that there were large numbers in this group of 2 and 3, and sometimes even more, where we would see large excesses of these loss of function variants. And we thought that was unusual. And so we decided to start by looking at those loss of function variants that were shared across all the populations that we had. And that's kind of represented here. Just focus on the red bits here, which is the ones that are shared by all the groups. And you can see it's a sizable proportion. And then when we took those variants and said, well, what genes are really being involved in here? There are, of course, a large number, but there are an interesting group that were all part of metabolic genes. And so these are genes that cause severe metabolic Mendelian disorders. So rare disorders, often in a recessive fashion. But you can see that the lightest yellow here is at 25% allele frequency. So you can see that some of these supposedly really damaging kind of variants are occurring at levels that would not be consistent with how often you see these kinds of disorders. And it's probably that they actually don't damage the protein in the way that we think, and they probably have a new function. But we thought that this may be relevant to the way in which sort of clinical geneticists think about these kind of loss of function variants. And so we started to then say, well, let's think about those variants that we know are medically relevant. And so the first list we went to is put out by the American College of Medical Genetics and Genomics. And they have a list of 58 genes where they say, if you see a variant in these genes, then this is likely to be causing disease and you need to sort of like act. And in our populations, this was very uncommon to see this kind of variation. Conversely, if we took variants that instead have been curated, so people have said, oh, I've looked at this variant very carefully, and I think that this is pathogenic or is highly likely to be pathogenic, we could see that almost everybody carried at least one of those variants, and on average, people carried like 7, these very deleterious kind of variants, which we thought, again, was a really unusual kind of setup. And so we said, well, what are these variants and how are they distributed? And so this is what you see here. You see that some of these variants are occurring at frequencies that are almost 50% and higher. And so to just give an example, here is a variant in LDL receptor. So this is a receptor for familial hypercholesterolemia. It's a recessive disorder, but the frequency of this variant in the existing databases is pretty uncommon. So less than 1%, which should be consistent with a recessive inheritance, and low even in sort of big databases just looking at Africans. But in our populations, we're seeing this thing at sort of 10, 12% in at least one population. And in fact, when we looked at it across the population, it's actually in 2 quite disparate populations. So in Botswana, it's at 12% thereabouts, and in Cameroon, a similar kind of 9 or 10%. So our interpretation is that, this supposedly rare variant, this may not actually be a damaging variant. You just had to look in the right populations in order to decide this. And so given that, one of the ways that clinical geneticists work in terms of thinking about whether a variant that's observed on sequencing is likely to cause disease or not is whether it's very rare, since the disease is very rare, then we thought that may help to sort of redefine and recharacterize some of these variants going forward. And that's something that we've done with various groups. So this all came together then when we looked at medically relevant variants that are relevant in the African context in particular. And that includes things like the sickle allele, which is here in orange, the APOL1 alleles, which cause a severe, predisposed to end-stage renal disease. And you can see their distribution here across West and then Central and then East Africa. But once you get outside of those areas, you really don't see it very often at all. And this is because these equatorial areas have very high frequencies of things like malaria, and which drives the sickle allele to be more common, or trypanosomiasis or sleeping sickness, which drives these APOL1 alleles to be more common. And so it does vary a lot depending upon where you are geographically. But even within a geography, we could see some variation again. So these are 2 groups, both in Uganda. And you can see that at these medically relevant variants, they differ at almost every single site in terms of the frequency of the variants in the 2 groups. And this is because they are separated by ancestry. So the UBS are the Bantu speakers. They are very close to this Niger-Congo group. And you see that on these principal components. Whereas the Nilo-Saharan group migrated down from Somalia. And so they are more in keeping with the Nilo-Saharan group on this side. And you can see that variation, so same country, but very different ancestral background, creates a lot of the variation at the disease susceptibility level. Okay, so that's a good start. And since that time, there have been these compilations of the sequencing that's occurred, and certainly although H3 Africa is still the largest, is the lion's share of the sequencing that's been done, the largest dots here, there's still more sequencing that has been done across the continent, which is good. But it's always good to bear in mind that Africa is a very large continent. It's misrepresented on most maps, and almost all the United States could fit within Africa, as well as most of China and India. And so if you think about the 2,000 ethno-linguistic groups across the continent, you realize that we're really just at the tip of the iceberg here. And so that's one of the reasons why there's been these calls to have much more sequencing, because there's so much to be gained, not just for Africa, but globally. Okay, so I'm going to switch gears here a little bit and sort of try and give an example of how this is playing out in some of the work that we're doing. So the first vignette has to do with pediatric HIV. And so this slide is really just a reminder that HIV is still a major global health burden with, 1.5+ million people newly infected each year. And this is also true when you talk about children who are living with HIV. And you can see the vast majority of these are in Western and Central Africa, and then Eastern and Southern Africa. And so that's sort of the major driver for the Collaborative African Genomics Network, which is one of the networks that I am still a part of. And this is a collaborative U54, so this is a funded grant within H3 Africa. And it consists of the university sites in Uganda and in Botswana, alongside the 3 clinical sites in Uganda, Botswana, and more recently in Swaziland, which is now known as Eswatini. And so the clinical sites are really the touch points for sort of recruitment and community-based sort of interactions. The universities work on the sort of genomics technologies and research, and then they actually send trainees who came to Baylor College of Medicine, spent 2 years working on the data for this before returning to their home universities, where they're sort of taking up faculty positions, now armed with human genetics. And so the major focus of CAFGEN has been HIV disease progression in African children. And so vertical transmission of the disease from mother to child in pregnancy has a varied outcome in terms of when you go from being HIV positive to having sort of full-blown AIDS in the absence of any antiretrovirals. And that distribution looks something like this. And so at one end, there are those individuals who progress very rapidly, rapid progressors. And at the other end, there are the sort of long-term non-progressors who can go 10, 15, 20 years without having any signs of developing AIDS. And so in the adult populations, genetic variants have been identified that sort of, have very strong influence on this dichotomy. The most notable one is a small deletion upstream of the CCR5 gene, which is a chemokine which the HIV uses to get into cells, and then HLA variants, which also modulate immunity. However, in Africa, there's no such known variation. And in fact, the CCR5 delta 32 does not exist. And so we were interested in seeing whether or not in our population we could identify variants of a similar kind of effect and character. And so to do this, again, we had our countries in Uganda, Botswana, and Eswatini, and we collected 500 individuals at either end of the spectrum in the first phase. And then in the second phase, which is just rounding up now, we've collected another 500 in that group. And we got blood-based DNA from all of these individuals, and then we did a number of different tests. So in some cases, we did DNA sequencing. Sometimes we did genotyping across the genome. And then we could use that data to do different analyses in order to understand the sort of architecture of the disease. And so today I'm going to talk about the genome-wide analyses and then a little bit of the HLA and virome analyses. So for our genome-wide association, one of the things that was been developed is a genotyping chip that's based on the sequencing data that I just spoke about. And that genotyping chip then we can sort of infer missing genotypes from the sequencing. And then we can do our sort of association testing. And so when we did that in our group, we get this kind of phenomenon. So this is just from the first sort of 1,000 individuals. And so again, in these Manhattan plots, each dot is a different SNV or a different variant. The higher the dot, the more significant it is along each one of the chromosomes. And so we have these cutoffs for genome-wide significance here in the red, and then sort of a suggestive significance in blue. And you can see that on chromosome 14, we had these two variants that were sort of just making genome-wide significance, and really were sort of like making more likely to be a rapid progressor. And you can see that these are sort of like equally distributed between the two countries. And in fact, this top variant is what's known as an expression quantitative trait locus. So the genotype actually influences the transcription of this PRORP gene. And PROP, in addition, is known to influence a number of sort of immune phenotypes, including leukocyte counts and myeloid white counts. So we think it's a really good candidate as something to take forward. And as we're doing our second phase of genotyping, we're hoping to sort of validate this early discovery. But you'll also notice this site here on chromosome 7, which is in an intron in this gene, which I'll come back to. And this is much more, again, more frequent in the rapid progressors. But you see here, it doesn't quite reach significant because although it's highly significant in Uganda, it's almost not significant at all in Botswana. And in fact, if you just look at Uganda alone, this same site comes up to be highly significant with a number of other sort of trailing variants that we see, in addition to a locus on HLA. And so, you know, in one sense, this is actually a good candidate because MAGI2 also goes by the name of AIP, which is part of the ALEX complex, which is involved in the way the virus hijacks the human machinery to be able to sort of bud off and be released. So it's really a strong candidate to be involved in this process. But the really striking thing that came home to us is that, you know, we're dealing with two different countries where the genetic variation and background is very different, and they have different strains of HIV as well. And so this might be the primary driver as to why we see, you know, a strong association of this locus in Uganda, but don't see it at all in Botswana. And these are some of the kind of challenges that we have to work through. But we could also say, well, let's talk about coding variants, right? So again, we're going to start by looking for these sort of big effect coding variants, and then sort of see if we can replicate it in a subgroup. So this is again from the first phase of data. And the first thing to say is that we confirm that CCR5 delta 32 is not observed. And in fact, if we just take all the chemokines and all the chemokine receptors, we don't see anything at all. So a very different pathophysiology or immunology for pediatric HIV than adult HIV. So what we decided to do was to start by taking the first third of the data set. They all had whole exome sequencing, so focusing on sequencing the coding regions of the genes. And when we did that, we found a variant which is a novel variant. It's not seen at any appreciable frequency in the 1,000 genomes or in any of these kind of big databases. But you can see it's highly enriched in our rapid progressors, but not seen at all in our long-term non-progressors. And so we were like, whoa, this is really interesting. And it was even came home more because we looked and saw that this coding variant is actually expected computationally to be a very damaging variant to this gene. The gene itself has strong links to immunity. And so we thought, wow, this looks really promising. But then when we genotype the rest of the cohort, turns out that this is just a common polymorphism, right? So this is one of those variants that is, you know, said to be damaging, but occurs and is very novel, but occurs at a very high frequency in this population. And we had a few more like these, which are all very disappointing. And so now we've taken a new view, which we're going to sequence all of the individuals in that first 1,000 batch, and then use our second phase of sequencing to sort of back it up. But there's still other things that we could look at. So from the first phase, we also looked at HLA types. HLA, as I said, has been really shown to be important in HIV. And so we could identify a number of HLA alleles from the sequencing that were highly enriched in both of our cohorts. And those are the ones that you see here. And whereas these variants are generally sort of 1% in the sort of general population, you can see that they're sort of approaching 20-25% in our populations. And that was suggestive to us that maybe, and this has been noted before in other groups, and it suggests that maybe there's a set of variations that have to do with whether you get HIV or not. And those might be completely different from the variants that may be important for whether you progress to your disease faster or not. So we did look for variants that differentiate between our two progression extremes, and we found 4 loci at HLA B and C. And you can see there are 3 B loci. The shaded ones here are loci that are more susceptibility, and then the open one here is more sort of protective. And so these loci had pretty decent effects. And these effects are also consistent across Botswana and Uganda. That's the ones you see in red here. And the protective effect was also consistent across the two population groups. So good internal replication. And when we looked in the literature, we could see that although many of these alleles, the B alleles in particular, have been associated with protection from HIV in adult cohorts, this HLA-C allele has not been, except in a small cohort of adolescents from Zimbabwe. So, we're thinking about this as a variant that might sort of say that there are, again, these differences between how childhood HIV progresses versus adulthood. And that has implications for sort of attempts to make vaccines and epitopes. And cumulatively, we think the HLA might account for maybe as much as 10% of the variance in progression. And we'll see whether or not that pans out as we get larger sample sizes. The other thing we could do is that we could take this sort of coding data, and instead of looking at the variants that are mapped to the genome, right, so mapped to the coding genome, we could say, well, what about all the things that didn't map? And could we find some information there, specifically looking for viral reads among these unmapped genes? And so this is kind of a schema that illustrates this. You can see that the vast majority of the reads will map, that's in green, to the actual exon. A lot of them will map close to the exon, so that's in the intergenic regions. But then there is this very small portion, which is blown up here, we'll say 1 to sometimes less than 1%, where you don't have any reads that have mapped. And you think, well, that's a really small percentage, but you have to remember there's sort of millions and millions here. So there's still millions of reads that you can interrogate here. And so from these reads, we're actually able to identify 42 different species with a pretty good identity. So as high as 85% similarity with specific species. And when we looked at those species, and this is kind of like a sanity check, right? We sort of look at how they distribute in the two different populations. And we can see that in general, you see a lot more of this in Uganda. Uganda tends to have a lot more of sort of infectious burden relative to Botswana, which is in the south. But you could also do a sort of additional sanity check, right? So for instance, this Toga viridae group is represented by the so-called Semliki forest virus. And sometimes that's seen as a contaminant. But in this case, the Semliki forest itself is actually in Uganda. And so we think that this is probably real. And it represents, because both of the groups were processed in exactly the same way, and just sort of represents the burden that one has in that context. So then got to the meat of the matter. So we said, well, what about in our progression groups? Do we see an excess of sort of viral loads in one group versus the other? And certainly we see that evidenced here by a higher proportion in the long-term non-progressors. which was statistically significant. And actually, if we then sort of like play out what's actually driving that, it turns out that almost all of it is being driven by this Aniliveride group. And Aniliveride, this Aniliveride association is actually quite consistent, whether we did it by gender or we did it, by different countries, whether we did it depending upon the sequencing platform, it was always fairly consistently significant. And so, like, what is Aniliveride? So most of these belong to the Torquay-Tenovirus group. It's not known to be pathogenic, but it does have strong interactions with T lymphotropic viruses like HIV, suggesting that it may be involved in sort of immune quiescence. And immune quiescence is very much the sine qua non of the early part of HIV infection in kids in particular. And so sort of Savannah, who was the grad student, sort of developed this kind of schema for this, indicating that there are many ways in which Aniliveride could act, but a primary one would be in the quiescence of the NF-kappa B pathway, and therefore facilitating sort of quiescence of the HIV machinery. Now, the problem with that is that by the time you're an adult, most people are infected. It's like CMV or any of these kinds of things. So it's difficult to know exactly what to make of it in a childhood context. What we did find is that the frequency that we found in our LTNP adolescents was actually more than we saw in an adult group who had whole genome sequencing in Uganda. And so we think there might be some relationship, but of course it's hard to tell whether this is cause or effect, right? So do they have this accumulation because they're HIV positive and on antiretrovirals, or do they have this accumulation as are they progressing because they actually have Aniliveride? And so this is the kind of thing that requires sort of more model-based studies, but at least gives us, again, as a discovery, an in to be able to do this. Okay, so I'm going to move now to the last vignette, which is something that I know that the medical students are quite familiar with. But I think it sort of illustrates this idea of how the genomics is relevant on this side of the Atlantic, even though we've been talking about Africa for the most part. So this revolves all around sickle cell disease, which is the most common single gene disorder in the United States. It is a recessive disorder, and the most common cause is a single point mutation in the beta globin gene, HBB. And it has a carrier frequency that's close to 8% among African Americans, but it's actually very common. It's also commonly seen in lots of population groups and ancestries. And so in African Americans, it can have a frequency that's significantly higher. So the primary way in which it causes disease is by causing this sort of sickling of the red cells. So the beta hemoglobin does not form properly with the alpha hemoglobin. You get polymerization. And so instead of these nice, slippery, sort of ovoid looking red cells, you get these kind of sickled red cells with the polymerized hemoglobin. And often these will interact with the endothelium to cause problems. They get stuck in the vasculature. And so basically anywhere that blood goes, you can get problems. So that runs everything from stroke and cerebral infarct all the way down to sort of bone problems and ulcers in the foot. Now, as is well described, the overall prevalence globally of sickle cell disease is most commonly in Africa. And this, of course, is because of endemic malaria as the primary driver, and that individuals who are heterozygous for the sickle allele are often protected from severe malarial disease. And so sickle cell from there, via forced and voluntary migration, then made its way around to the rest of the world, so that now there's a very broad global prevalence attached to it. And one of the staples of sickle cell disease management in the United States is the transfusion of red blood cells. And as the gene therapies and other sort of curative forms are coming on board, transfusion still remains a staple until that's widely available. But transfusion is not without its problems. So somewhere between 6 to 9% of sickle cell patients will develop what are called alloantibodies after transfusions. And so these are antibodies that are developed in response to antigens, but minor antigens. So all blood is going to be matched for the major antigens, which are A and B and D, which is rhesus. But there are these minor antigens, C, E, Kel, etc., which most people don't form any substantive reaction to. But in some group of individuals, when they see that mismatch between the donor blood and their blood, they will develop these antibodies. So this is like 10% of individuals. And we generally refer to these individuals as kind of responders, as opposed to non-responders who don't make it, which is the majority of individuals. And so this is a challenge for responders in that if every time they develop an antibody to one of these minor antigens, then the next time you have to sort of type their blood to ensure that they don't receive that antigen. And so eventually you get to a point where you've just sort of exhausted all the blood that you could possibly get your hands on. And if they get incompatible blood, then they run into the problems of hemolysis. So this is a real challenge in the transfusion medicine world. And so we were fortunate to work with a couple of transfusion medicine physicians from the Gulf Coast area to conduct a genome-wide association study of this sort of responder status. You can see the numbers here. And again, these are all individuals who were collected in this sort of Texas, Oklahoma, Arkansas kind of basin. These individuals were all genotyped for 2.5 million variants across the genome as part of this genome-wide association study. And when we did this, there were 2 sites that got not quite to genome-wide significance, but close enough, one on chromosome 5 and one on chromosome 2. And you can see that when we then sort of imputed data, so we sort of like make a statistical guess as to the missing genotypes, we can see that we get a little bit closer to the magical genome-wide significance cutoff line. And so we decided to continue looking at these 2 just because we know that these genotyping chips don't actually cover, or imputation doesn't actually cover African populations particularly well. So we were mindful of that. And so we were fortunate to get a sort of second cohort that was independently collected and had multiple transfusions. They had mostly sickle cell, but some had some other hemoglobinopathies, but the same criteria for calling somebody a responder or a non-responder. And when we do that, you can see that, although it's a smaller data set, we still see the same sort of general view of the frequencies, and the odds ratios are all in the same direction. And so we're now sort of at that genome-wide significance when they're meta-analyzed. And then what was really interesting is that the people who are really problematic are the people who make lots and lots of antibodies. So if you get beyond the C, E, and Kel, they're still making antibodies if you match for those blood. And those individuals, again, they were the primary drivers for this association. So this is telling us that this is more about, not so much about the mismatch, but about how you respond to the mismatch in the donors. So we looked at our variant, our best variant here. You can see its frequencies in the cases and controls, and you get this sort of large effect. The frequency in global populations is, again, sort of more consistent with what you get in the controls. But interestingly, when we broke down those populations, you could see it's much more common in individuals with West African ancestry than European ancestry. And so we were sort of mindful of the fact that most African Americans have some measure of admixture of European ancestry. And so it could be that this was sort of confounding our association. Now, we do try and control for that. Again, using these principal components, we try and control for the sort of global overall ancestry of individuals. But that doesn't really tell you anything about what's happening at the local chromosome level. So here, the green and the blue represent, again, sort of European and African ancestry. And you can see that it really depends upon where you are in the chromosome as to what your true ancestry is at that point. So we tried to leverage this to sort of better understand our association. So on average, African Americans have about 22% European ancestry. It's a little bit lower in our sickle cell cohort, but we found, pretty close. And so we could test this business of using sort of local ancestry across chromosome 11. So here you can see this is a proportion of African ancestry as you move from one end of the chromosome to the other end of the chromosome. Here is a genome-wide average, which is about 17%. And on chromosome 11, at this particular locus here, almost everybody, cases and controls, are 100% African. And that's because this is where that beta globin locus is. And as we said, the sickle allele sort of generated in response to malaria in Africa. And so we could leverage this kind of local ancestry to do our association. So now we restrict our analysis depending upon whether a particular locus has more African or European ancestry. And when we do that, we can see that in the African ancestry, we see a very strongly significant signal at our chromosome 5 locus, which is not evident when we do the analysis depending upon your European ancestry. And in fact, as we've looked around, that variant has varying frequencies across Africa, but to date, we have not seen it in any of the European populations. And the reason that's significant is because there was always this idea that the mismatching itself, and the reason that individuals with sickle cell sort of have a lot more alloimmunization was because they were mismatched. But this suggests that maybe the reason that they have a lot of the higher prevalence is just genetics and maybe not the mismatching per se. And there's some epidemiological studies that back that up as well. So where is this locus, right? So this is on chromosome 5. It's on chromosome 5q33, which is actually a locus that has a lot of immune factors. But the particular variant itself is right over this enhancer region, has multiple transcription factors and open chromatin, and is actually known to influence the expression of the two neighboring genes, which we know not that much about, but there's a link RNA or non-coding RNA that's highly expressed in the liver. Liver is good because it is involved in sort of erythropoiesis, one of the sites of extra outside the bone marrow hematopoiesis. The other gene that's there is adrenoreceptor, again, highly expressed in the liver and in the spleen. So again, the spleen is another site of extra poetic red cell production, so a good candidate. And in fact, when we looked more closely at where the protein that's being transcribed from the adrenoreceptor is, it's in lymphocytes in the red pulp, so it has a direct connection to the red blood cells, that particularly old red blood cells that are brought into the spleen for destruction. So really good candidates for sure. But shortly after that, we got a phone call from a transfusion medicine person who had heard this presented at a meeting, and he said, "Oh, I think you got the wrong gene. I think it's really this gene upstream, which is known as IL-12B." And actually, he was able to show that in primary B cells, there is this 3D conformation of the DNA that puts the IL-12 in contact with this enhancer region. And you see that sort of illustrated here. So each one of these red points here is points where the DNA is folded onto itself. And you can see that within this one region, the GWAS enhancer is now brought sort of physically closer to IL-12, suggesting that there might be some coordinated transcription across this. And IL-12B is also interesting because it's known to be important in dealing with intracellular organisms, particularly things like, this is Mycobacterium leprae, but it also works for Mycobacterium tuberculosis. And so the locus itself has actually been associated with resistance to tuberculosis in East African populations. And so we're sort of working now on the hypothesis that you might have this kind of pleiotropy between like resistance from tuberculosis, but more susceptible to alloimmunization. And I think that's going to be a little bit of a theme as we move these things from Africa and across the Atlantic. We certainly see it with beta globin and the sickle allele, where you sort of protected from malaria, but then you get sickle cell disease. With APOL1, where it is susceptibility to end-stage renal disease, and you can see it's like a 10 times higher relative risk in the US populations, but highly protective from trypanosomiasis in the African context. And then more recently, a gain-of-function mutation in the mechanoreceptor causes iron accumulation in adults. These are mostly African Americans. But in the context of Africa, again, sort of resistance from malaria, and it's particularly common among individuals of African ancestry. And so this idea of sort of transatlantic pleiotropy, I think, is going to continue to play itself out. So that's really all I had. Hopefully I've convinced you a little bit more about the importance of doing genomics in African ancestry populations and how that has relevance to human health and disease mapping from a human genetic standpoint. I always have to thank individuals. Been incredibly blessed to work with individuals in H3 Africa. They did most of the work. I just happened to be on this side of the Atlantic, so I tend to give the talks on this side. But really, this was really driven by African scientists. And then the trainees from CAFGen who did a lot of the alloimmunization work and did all of the HIV work when they were here at Baylor, as well as collaborators, and then the lab, who have been sort of incredibly supportive in this journey, and the funders as well. And then with that, I am very happy to take questions.
Thank you so much for a really interesting talk. We have a few questions in the chat, and then if there's any questions in the room, or feel free to jump in. Our first question comes from Dr. Anthony Lamantia, who actually has a couple of questions, but thinking about the idea of variants that are more, that are identified maybe by computationally as being damaging, but then show up at higher frequencies. Dr. Lamantia asks, are damaging variants modulated by background variation? In other words, if a variant is disease associated in a European background, it might have the same effects on the protein, but other interactors modify and diminish the deleterious effects.
Yeah, so I get that question a fair amount, and it's a great question. In that, for instance, with that familial hypercholesterolemia variant, could it be that it's sort of maintained, even though it is actually damaging and pathogenic? Is it just sort of maintained there because of other background factors, or some other kind of pleiotropy factor or adaptive factor? And the answer is I don't know, but it certainly could be. And it would be an interesting story if that turned out to be the case. In some senses, we found so many of these things that I can't imagine that that's what's occurring all the time. I think sometimes the simplest explanation is that it's just the sort of misclassified because it's not really classified with the sort of full information available. But I think that there are going to be these instances where seemingly pathogenic variant at a very high frequency doesn't cause disease, but there's some other kind of factor that's sort of playing a role in its frequency. Teasing that out is going to be difficult, but I think it certainly could be playing a role.
Great. Another question for Dr. Lamantia. So there's data that, at least in the US, health disparities are less about biological variability and disease risk, and more about sort of delivery and access of care. So is the genetic variability that you see consistent with that?
Yeah, so that's a really important point, and I should be really clear about that. That I think that the vast majority of health disparity that you see in terms of outcomes for chronic diseases is the lion's share of that is a result of environmental variation. I have little doubt about that, and there are good examples of very simple interventions which have made big differences to health disparities. So I do think that there are going to be some things where there is a big genetic effect that plays a role in it. But I think that for the majority of things, the genetic role in terms of the large, stark health disparities that we observe is going to be relatively small. And that the thing you need to fix for the majority of sort of well-established large health disparities is the social and behavioral determinants, and that understanding these genetics is more about understanding the basic biology of specific diseases and how you can potentially intervene therapeutically with these specific diseases. But I think on the context of health disparities, there's a lot of confounding, but the majority of it is legacy based.
Well, I'm going to give our students in the room an opportunity, if they like to, come down and ask a question rather than me try to translate it across the microphone. But in the meantime, while people are thinking of those questions, my question is more about the work that you are doing is really generating so many different lines that could be followed from a research standpoint. So every variant that is identified as potentially, modifying disease impact is something that a thread that could be followed up. So as a director, as the head of the new institute that you are sort of leading up, what is the strategy? How do you ensure that some of these stories get followed up, and how do you support scientists, and how do you pick which ones to follow?
Yeah, that's the question. I think so. For me, I'm a lowest hanging fruit type of person. So I am very much interested in the ones that have the largest effects, and that the things that appear to sort of give rise to a natural line of experimentation that one could get to eventually. And so that's kind of how I pick things. But, as you do these things, especially as you do more of these diseases, you discover that you can't chase everything. And in some ways, what you'd like to see happen, which hasn't happened, but I'd like to see it happen more, is that the field takes on some of these things, in that, having done the discovery part, I feel like there's some things that I definitely want to carry forward into the next part, which is more digging into mechanism and trying to understand what's going on. But you can't do them all, and you sort of have to try and engage the rest of the field to take on things, which has been challenging in terms of trying to, convince people that there is some relevance here to the work that they're doing. But you can only do so much.
Well, I think you're filling the literature with a lot of places that people can pick up and follow up. So there is another question here in the virtual world. So Noor Tasneem asks, in 2019, Shmona Simpson published a comment on the Lancet Hematology titled "Sickle Cell Disease: A New Era," highlighting the sickle cell disease burden in children in Africa. Your talk discussed allele association with HIV disease progression in pediatrics in Africa. What future work will be done in sickle cell disease genetics to address its burden in children?
Yeah, so that's an excellent question, and timely, I think. So the NHLBI has actually invested a large sum of money to build a network of infrastructure to address sickle cell disease in both children and adults across the continent, so that there are sites that go from Mali all the way down to South Africa, much the same way that H3 Africa is built. It's called Sickle in Africa. And they have a pretty, deep mandate to be able to first kind of, build the kind of infrastructure and registry that one needs to address these. But I think that from an advances standpoint, the gap is that there are all these developments of, new therapies and new sort of gene therapies, et cetera, that are being developed, in the US and in the UK. But the real burden of disease, as you point out, is in Africa, and figuring out how well some of these sort of therapies and so on are going to translate over there, and how they can be translated in an equitable and cost-effective manner, I think is where there's a bit of a gap right now. We've done some work looking at the beta globin locus and cis modifiers of fetal hemoglobin in African populations. And there's a lot of variation and difference between what you see across the continent relative to what you see in the United States. And that has implications for, very specific targeted therapies that are, dependent upon your particular variant or particular site. And so I think that's a gap about how the plan is to translate these things across. But I think the idea of that the sort of funders have, and perhaps the field, is that you have to sort of build the infrastructure to be able to ask those questions first. We'll see. Great,
thank you very much. I think we'll do one more question from Asun Imani. Onset of COVID-19, at the onset of COVID-19, there were suggestions that mortality rate of the infection would hit Africa heavily. However, data do not support that. Do you think allele protection or variant gene could have contributed to the low reported infection and death rate on the African continent?
Yeah, that is a fascinating question. It's one that H3 Africa has kind of grappled with a little bit. I should say that H3 Africa has been very good in the COVID-19 pandemic because they've really, having the genomics infrastructure there has facilitated testing and diagnostics. So they're able to do that sort of sequence-based testing and sort of being able to do real-time sort of surveillance of the disease. So that's been good. But I think from the human side or the human genetics side, it's a little bit trickier. Initially, I thought that some of the lack of big heavy hit there, had to do with it's a very large landmass, and that some of the environments, particularly outside of the urban areas, would sort of allow for less crowding, and that even within the urban areas there's a lot more sort of open air and a lot utility of cross ventilation, that kind of thing. But of recent, over the last, in this last wave, there's been a lot more in Africa. It's picked up, and that could be sort of variant related, but it's definitely picked up, and it's made people think about, well, is it all, sort of non-genetic, or is there something there? There are a couple of studies that are contributing to the very large human genetic consortia that are looking at sort of host susceptibility. They have not found anything that is different from what's been seen for any of the other groups. But I think that the data is, there's not as much data, and it's sort of somewhat behind. So we'll have to see. My answer to that has changed over the course of the pandemic, but I'm hopeful that we'll get an answer.
Great. Well, thank you so much. We really appreciate you sharing your talk with us and spending some time with us here today, and hope to communicate with you again soon. Thanks so much.
Thank you for having me.